So now we'll be studying the concept of deamination. Now whatever food we eat, it has proteins, fats, carbohydrates, and other nutrients. So the carbohydrates are broken down generally to glucose, and excess glucose in a body, which is not used, is converted to glycogen and stored in the liver and muscles. Now, extra fats is stored as adipose tissue. But if you have extra amino acids are usually not stored, they are usually removed from the body. So, the general process, what happens is basically in our tissues, if there is extra amino acids, what happens is through the process of transamination first, what happens is the amino acids convert to a central molecule like glutamate. So maybe an amino acid like alanine or aspartate, they will get converted to glutamate by transamination. And then the glutamate goes into the liver, where in the liver mitochondria, hepatocyte mitochondria, deamination occurs. Now, this deamination leads to the liberation of the amino group from the glutamate. That amino group becomes ammonia. That ammonia enters the urea cycle. So, this is how the body takes care of the extra amino acids present and eliminates the excess of ammonia in the form of urea from the body. So, what is the definition of deamination? Deamination is the removal of alpha amino group from an amino acid to form a keto acid. So, you're removing the amino group of the amino acid to form a keto acid. And what is the general reaction of this? Generally, the structural representation of this reaction is like this. This is, if you can recognize, it's the amino acid. It has the NH3 plus group, it has the COO minus group, it has the H, and it has the R group. Now, usually you need water molecule for deamination. So, the amino acid combines with the water molecule, hydrolysis happens, enzyme is involved. We will talk about the enzyme now, and this leads to the liberation of ammonia. Ammonia, this will enter the urea cycle, and from this, if you remove the amino group, you get a keto acid. So, as the definition goes, removal of the ammonia group to form a keto acid. So, deamination can be classified into two types. One is the oxidative and the other is non-oxidative. Now, oxidative deamination is always kind of a link between the amino acid metabolism and carbohydrate metabolism. We'll see in a while. Let's see some features of first the oxidative deamination. Oxidative deamination occurs in the kidney and liver cells. Okay, the enzyme that is involved is very important. It is called as L-glutamate dehydrogenase. LGDH, short form. L-glutamate dehydrogenase. So what is the reaction that is happening here? You can see the reaction. So you have L-glutamate here. L is because the amino group is on the left hand side. So you have glutamate with its side chain as CH2CH2COO minus. Here, very important cofactor comes in, which is nothing but NADP plus. Not NAD plus, usually NADP plus, even NAD plus can also come. So what is NADP plus? Nicotinamide adenine dinucleotide phosphate. In its oxidized form, NADP plus, it will come and it will first oxidize L-glutamate into alpha aminoglutarate. So what is happening here? If you see this CH and NH3 plus, the amino group is being converted to C double bond NH group. Now this group is called as an imino group, not an amino group, but an imino group, IMINO, imino group. So this is an unstable intermediate. So the first step in the process of oxidative deamination is L-glutamate with the action of NADP plus and of course glutamate dehydrogenase enzyme. NADP plus gets reduced to NADPH and H plus, and L-glutamate becomes alpha aminoglutarate. Okay, and in the next step, hydrolysis happens where a water molecule enters the reaction, and from alpha aminoglutarate you get alpha ketoglutarate. This is the structure of alpha ketoglutarate. Now, if you see, the final product of oxidative deamination is alpha ketoglutarate. Doesn't this look familiar? Isn't this a very important intermediate of Krebs cycle? So, from excess amino acids, transamination is happening, and then you get glutamate. Then glutamate undergoes this process of oxidative deamination and produces alpha ketoglutarate, which is a very important intermediate of carbohydrate metabolism, Krebs cycle. So, this oxidative deamination, this whole reaction is kind of a link between the amino acid metabolism and the carbohydrate metabolism. Now let's study some basic features of this enzyme, that is glutamate dehydrogenase. First, about GDH. The first point is that it is a zinc containing metalloenzyme. So it requires zinc ions to function properly. The second is that it is made up of 6 subunits. It is a hexamer. It is made up of 6 similar subunits. The third point to remember about GDH is that it is present only in the mitochondrial matrix. So this reaction that you're seeing here is happening only in the mitochondrial matrix of hepatocytes, that is liver cells and kidney cells as well. So it is present only in the mitochondrial matrix. The fourth point is it is an allosteric enzyme. So the regulation that happens for this enzyme is always allosteric. The next point is ADP and GDP are positive regulators. Now, we can think about it like this. If ADP and GDP are present in excess numbers, that means if ADP is in excess, that means ATP is less, because ATP has been broken down to form ADP and PI, right? So if ADP is more, that means ATP is less. And if ATP is less, it is an indication to the cell that energy is lacking. We need to produce more ATP. So what does the cell do? The amino acids are converted to glutamate by transamination. Glutamate undergoes this process to form alpha ketoglutarate. Alpha ketoglutarate will enter the Krebs cycle. This will ultimately go for oxidative phosphorylation to produce ATP. So ADP and GDP, of course, are positive regulators. They are indicators that ATP and GTP is lacking in the cell. So it guides this enzyme to carry this reaction in the forward direction, and L-glutamate is formed by transamination, and you get excess of alpha ketoglutarate, which then helps in production of energy, that is ATP. And of course, the last point is ATP and GTP are negative regulators. Similarly, by the same logic, if you think about it, ATP and GTP are excess in number, then ADP and GDP are less. That means if ATP is sufficient, which means the cell has enough energy to function, so this reaction does not take place at a very fast and quick rate. It slows down for a while unless the balance is restored.